Good morning. Today we'll be talking about Chapter 6, the first part in particular, but Chapter 6 is going to cover the cartilage and bone connective tissue. The first part that we'll be looking at today is going to be the cartilage connective tissue. Some general characteristics of cartilage connective tissue is it's weaker than bone, but it has more resiliency and is more flexible to stress. Some important factors or functions of cartilage include supporting soft tissues, gliding surfaces at articulations or joints, and also provides the rough draft for bone formation when we get into endochondral and intramembranous ossification. So when we're looking at the types of cartilage, there are three main types of cartilage in the human body, and we'll start out with the first one called a hyaline cartilage. The hyaline cartilage, as mentioned, is the most abundant. We find this in the articular aspects of the joints, so we can find those in the shoulders, in the ankles, in the elbows, in the carpals of the wrist, and also in the phalanges. Some of the things that make hyaline cartilage interesting is that it's very flexible and resilient. It takes on this translucent color or translucent appearance as shown in the blue in the diagram. And it's also surrounded by a dense connective tissue called a perichondrium. And then a perichondrium is going to have two distinctive layers, which we'll get into here in a second. The next one that we look at is going to be the fibrocartilage. The fibrocartilage is mostly resilient to tensile or stretching and a lot of compression forces. So with fibrocartilage, we find this in the pubic symphysis. We also find this in the cartilage of the intervertebral discs, and we also see it in the meniscus or the cartilage pad in the knee joint. And finally, the last one is going to be the elastic cartilage. With the elastic cartilage, it has highly branched elastic fibers, shown here in the slide. We can see a highly branched elastic fibers. It's highly flexible. It is surrounded also by a perichondrium. And with the surrounding of the perichondrium, we also see this located in the cartilage of the nose. We also see it in the ears. We also see it located in the epiglottis. Some of the key components, or just as a review when we looked at the histology of cartilage, is remembering that the lacunae forms or has the individual chondrocytes. The chondrocytes are encased inside of the lacunae and maintain the matrix or the extracellular matrix. So when we're looking at the formation or growth of cartilage, a couple of key points is understanding the first one, which would be the interstitial growth. Interstitial growth simply means from within. What we're going to see is that the chondrocytes are going to undergo meiotic cellular division. So in the first slide, we see that the chondrocytes are housed within an individual lacunae before they actually begin their division. When we see the division in step 2, they are now considered to be a chondroblast once they start dividing. These chondroblasts are going to occupy, as it's shown here, its own single lacunae. So we see a chondrocyte in step 1 begins the division inside the mitosis or the division inside of its own individual lacunae, and then we consider this chondrocyte now to be called a chondroblast. When we see that, what we're going to see in step number 3 is we're going to see the secretion of a new matrix. That's the most important aspect in step number 3. These secretions of new matrix are actually coming once they have divided, they return back to a chondrocyte. And they are eventually going to reside in their own lacunae. But in step number 3, what we see is each cell is now considered to be its own individual chondrocyte. So in step 4, the cartilage continues to grow internally. We see each individual chondrocyte developing its own new matrix, and then we will then see these chondrocytes possibly returning to step number 1, beginning the division all over again to produce more chondrocytes. So when we're looking at appositional growth means the outside edges are the peripheral or the periphery. And the most important cell at the beginning in step 1 is going to be the mesenchymal cells. So the mesenchymal cells are located on the peripheral aspect of the cartilage. And what we're going to see is the division. So the mesenchymal cells are located on the outer shell, or what is known as a perichondrium. So we're going to see these divide, and they are going to divide into what we consider to be 2 different cells. One, they're either going to turn into divide into an undifferentiated stem cell, or they're going to divide and form another mesenchymal cell. Remember, an undifferential stem cell simply means that it won't have a function until it is located inside of the matrix. So once we see the undifferentiated stem cell divide, we go down into step number 2. So again, we have a undifferentiated stem cell. We have a committed cell that has changed into a chondroblast, and these chondroblasts are going to be secreting its own matrix, and it's shown in the slide of creating a shadow on the outside. They are going to be migrating eventually, as we see in step 4. Once they push apart, then they go back to being called a chondrocyte. So this new matrix that is being produced is going to occur throughout the lifespan or growth of the cartilage. We see interstitial and appositional growth decline as rapidly as cartilage matures because eventually the cartilage is going to become semi-rigid and the matrix is no longer able to expand. Once we see that cartilage has matured, no new cartilage can begin. So this is simply the growth phase of cartilage, and once we get into maturity, then we won't see any more of the division through the mesenchymal cells, and we will no longer see any more of the chondrocytes producing matrix. So in step number 3, what we have are just basic classifications, or slide number 3, what we have is basic classifications of bones by shapes. There's over 206 major bones in the human body, and their primary functions are either they're going to store minerals and energy reserves. 90% of our calcium reserves are stored in our bone. It is also a process for hemopoiesis, which is the production of red blood cells that are released from the bones, usually stimulated by either hypoxia or ischemia, which just means temporary loss of oxygen. Bones are also very important for leverage and movement. They provide attachment sites for muscles, soft tissues, and organs. And their job also is for support and protection. So we see this with the rib cage protecting the heart and lungs, or another example would be the cranium protecting the brain. So there's 6 major different types of bone shapes, and each one has a particular function also. When we look at The first one, we look at this in depth, is going to be the long bone. So this first one, the long bone here. The long bones, we see this in the arms, we see this in the femur, the humerus, we see these in the fingers and the toes. The next one would be the flat bone. Flat bones are thin, roughly parallel surfaces. They form the roof of the skull. We see this in the sternum also. We see this in the rib cage, or the ribs are considered to be the flat bone. And then on the posterior surface, we see the scapula is another example. The sutural bones, we can't really see these in the illustration, but the sutural bones are going to be the sutures that we see, like the frontal suture or the parietal suture. That helps divide the flat bone or the cranium. Irregular bones, a good example would be the vertebrae. The vertebrae, their most important functions is the more projections that we see in an irregular bone, the more opportunity for muscle attachments, especially when we look in the posterior surface, such as the erector spinae. We'll see multiple small different types of erector spinae muscles being able to connect either to the spinous process or the transverse processes. The next one, the short bones. Good example are the tarsal bones, or the bones of the wrist and the carpal bones. These are all considered to be short bones. And then the last one is the sesamoid bone, which is, I'll just circle it here, like the patella. You also have one in the base of your thumb, and you also have one in the base of your big toe. These help develop mechanical advantage. Tendons are crossing over these different types of bones. For instance, the patellar retinaculum goes over the top, which creates a mechanical advantage for the quadricep. The flexor, extensor, and abductor pollicis muscles of the thumb crossing over produces a mechanical advantage. So their prime job is free floating, but they produce a mechanical advantage for the muscles associated with that area of the body. So when we look at the general structure, this is an example of a long bone. We see, especially in long bones, we'll see a proximal epiphysis, largely composed of spongy bone. So you can see the lattice framework that occurs in the proximal epiphysis. Around the metaphysis, we see this is usually where the growth plate is located, between the epiphysis and the metaphysis. This is a transition, basically, the metaphysis, where we see this epiphyseal line showing mature bone growth, but it's a transition between the spongy bone and the compact bone of the diaphysis. So the diaphysis is composed mostly of a compact bone, and then again we have a transition in the metaphysis, and then finally the distal epiphysis. Also worth noting, in the epiphysis we see the articular cartilage. It'll be located in this example on the distal and the proximal ends. And this hyaline cartilage is important for articulation between the two joints. So when we're looking at the endosteum and the periosteum, we're going to start getting into some of the cells. First, we'll look at the endosteum. You can get to it. So the endosteum, we're going to be looking in the red bone marrow. So we have a periosteum layer, or an outer covering, and then we have a series of a compact bone, and then we have the endosteum. There's a blown up picture over here on the right side, And this endosteum is going to contain osteoprogenitor cells. It's going to contain the osteoblast and osteoclast. And we'll talk about each one of these individual, but these are active during bone growth, repair, such as breaking bones, or remodeling, such as like a hairline fracture. So the first one, the osteoprogenitor cells, these are going to be important because they maintain the population of the osteoblast. Osteoblasts produce new bone matrix in a process called osteogenesis. But the osteoprogenitor cells, they're important for repair or fracture. This osteoblast, they help make and release proteins and other organic components of the matrix. And they also assist in elevating local concentrations of calcium phosphate and promoting the deposits of calcium salts in the organic matrix. Basically, they convert the osteoid to bone. And finally, the last cell would be the osteoclasts. These have up to 50 different nuclei. So you can see here in this illustration, it's just showing you that this cell has a series of nuclei, but it can contain up to 50 different type of nuclei. This cell also, it's giving a little bit of an illustration. It goes through a process of osteolysis, which is basically releasing acids and protein digesting enzymes that secrete to help dissolve the matrix and release essential minerals such as calcium into the blood system. They're very important in regulating calcium and phosphate for the increasing these concentrations in the cardiovascular system. And finally, we'll look at the periosteum. This periosteum is composed of a dense, irregular connective tissue, and it has two different types of layers. The outer layer, or what is considered to be the fibrous layer, this contains the osteoprogenitor cells and the osteoblast. So this outer fibrous layer, that's going to cover the whole epiphysis through the diaphysis through the metaphysis, all the way around the bone. The fibrous layer, again, contains the osteoprogenitor cells and the osteoblast. This intercellular layer, it does give a little bit of an illustration. It has these anchoring collagen fibers shown here. These little anchoring collagen fibers are important. They're also called perforating fibers. And finally, there's a couple other important things that we should point out in this slide. We see an osteocyte here. This is a mature bone cell. It accounts for most of the cellular population within the bone. And these osteocytes maintain the protein and mineral content of the surrounding matrix. And the surrounding matrix is also called an osteoid. 1/3 of the bone mass is composed of organic compounds, and these organic compounds can be cells, collagen fibers, and important ground substances. So this is just an illustration of what we spoke about just for review. So when we see osteoprogenitor cells, they're differentiated stem cells. So we find these in the endosteum and the outer layer of the periosteum. These osteoprogenitor cells will then change into osteoblast, which form the bone matrix. Some of the osteoblasts differentiate into osteocytes, which are mature bone cells. And then over here on the right, as mentioned earlier, these osteoclasts, they can have up to 50 nuclei. Through the breakdown, they're releasing, the lysosomes are releasing the acid onto the matrix. You can see here, this is a osteocyte, another osteocyte here. Basically what they're doing is breaking down the osteoid and then allowing for those calcium and phosphate ions to be released into the bloodstream. This next slide is just a simple way of looking at the compact versus the spongy bone. This is an example of a flat bone, such as the skull. So we have the periosteum layer, or the outer shell. Then we'll have a layer of compact bone. It's usually compact bone is dense and solid. And then we have the spongy bone, or the diploid. And this spongy bone has a trabeculi, or what we consider to be an open lattice network. So there's going to be blood vessels that are going to be running in between all of this trabeculi. And then finally, we have another layer of compact bone, and then another layer of periosteum. So this next slide we're really going to be talking about is the osteome. So the basic compact bone is called an osteome. It's the basic functional unit of mature compact bone. Within this, we see that the osteocytes are going to be arranged in concentric layers around a central canal, or what we call a Haversian canal. So basically what we're looking at here is we're going to have a layer or a concentric ring that's going to be formed around a central canal. In this central canal, we're going to have the arteries, veins, and nerves that are going to be going up through this central canal, and all the osteocytes are laid down in a concentric ring around the central canal. So all of the nutrient distribution that the osteocytes are receiving are coming through the blood vessels, or the arterial flow coming up through the central canal, and then the osteocytes are going to be sharing that material throughout in the concentric ring through a series of connections called a caniculi. So these perforating canals, another name for them, perforating just means that they're moving laterally, so we can see the perforating versus the central canal, which is going more straight up. But these perforating canals are also called canals of Volkmann. They're extending, again, perpendicular to the surface. The blood vessels supply the blood to the osteomes deeper in the bone. It's also supplying the fibrous and cellular layer of the periosteum. So this is a little bit more of a blown up version that we see here. When we're looking at this, it's showing the concentric rings, and the concentric rings are called the lamellae. These lamellae are containing each one of these osteocytes, and then a blown up version shows the caniculi. So each one of these caniculi are going to attach to another osteocyte that's going to be in a central ring, and then we'll see it going around, attached to another osteocyte. So we can kind of see that pattern occurring in this lamellae or concentric ring. Again, to stress it, we receive all of the nutrients, the osteocytes receive all their nutrients through either the central canal or the perpendicular canal. So when we're looking at the structure of the spongy bone, The first thing is there is no concentric ring lattice network, so the lamellae are not arranged inside of the osteons. The matrix forms the strut or plates, and these are called trabeculae. And these struts or plates or trabeculae are basically formed by the matrix of the osteon. There's no capillaries or venules located inside of this. The nutrients reach each one of the osteocytes. So when we're looking at the osteocytes inside of one of the lacunae, inside of these trabeculae, the nutrients reach the osteocytes by diffusion. Through these caniculi are connections in between each one of the individual osteocytes. Most of the time what we see, the spongy bone, the location, they're usually located where bones are not heavily stressed or where basically stress arrives in multiple directions. This lattice framework, we find this in the epiphysis of long bones. It helps reduce the weight of the skeleton. That's its primary job, and it helps distribute the forces produced from such thing as walking or jumping. So that completes the first part of Chapter 6, Cartilage and Bone.